Good afternoon, everyone. Welcome to today's Canada C3 Hangout. My name is Joe Grabowski, and I'll be your host for today. A little background for those who may not know, Canada C3 is a signature project for Canada's 150th anniversary, and the centerpiece is an epic 150-day sailing journey from Toronto to Victoria via the Northwest Passage, with the goal of connecting Canadians from coast to coast to coast, and to inspire deeper understanding of our land, our peoples, and the past, present, and future of our country. So the expedition is divided into 15 legs, and we'll have a cross-section of Canadian society on board, including scientists, artists, Indigenous elders, historians, community leaders, youth journalists, and educators. Some may have joined us a few days ago when we connected to talk about microplastics, and that was during the 12th leg of the journey, and they'd just gone from the Bering Sea into the Pacific Ocean off the coast of Alaska. Today, I'm excited to introduce Julie LaRoche. She's a professor and Canadian research chair in the Department of Biology at Dalhousie University in Halifax, Nova Scotia. As a biological oceanographer, her research is focused on the study of marine phytoplankton and microbes and how they affect the cycling of carbon, nitrogen, and other elements in our oceans. So today's Google Hangout is entitled Tiny Critters, Huge Impact, and I have a feeling we're going to dive a little bit deeper into that today with Julie. So Julie, thanks so much for joining us today.
Bienvenue.
All right. Well, Julie.
Okay, I will start in English and switch to French to explain in both languages. Yes, so I work on plankton, and plankton are very small, but they're extremely important in the ocean because they're at the basis of the food chain. Donc, le plancton, c'est des petits organismes minuscules qui pratiquement flottent dans les océans. Donc, ils sont à la merci des courants marins. Et puis, ils ne peuvent pas voyager comme les poissons d'un endroit à un autre. Donc, the plankton, how small is it? So when we're talking about minuscule, tiny cells, we're talking in the size of microns. So 1 micron, if you look at my pen here, I don't know if you can see this. Okay, you see the point of my pen here. So this is about 1 millimeter in size, just like the very tip here. So when we're talking about plankton and bacteria in the ocean, we're talking about 1,000 of the size of the point of my pen here. So we're talking about very small creatures that we can only see with the microscope. These creatures, they are really abundant in the ocean. So, quand on pense au plancton et la grosseur du plancton, il faut regarder soit la largeur d'un cheveu humain ou la pointe de mon crayon ici, de mon stylo ici, je ne sais pas si vous voyez ça. Donc, on parle de cellules qui sont la grosseur de 1 millième de la pointe de mon crayon ici. Donc, très minuscule, mais très abondante dans les océans. Donc, dans une goutte d'eau, dans un millilitre d'eau, il y a environ 1 million de bactéries. So, in 1 millimeter of water, 1 milliliter of water is about 1 million of bacteria. So, when you think about bacteria, you think about probably something that's not good for you. So diseases like stomachache or something like that. But in fact, most of the bacteria in the marine bacteria in the environment and on your body as well, most of the bacteria are beneficial to us. And in the ocean, it's the same thing. So, so I'm going to go through the different types of plankton. So when we're talking about plankton, or we're talking about the plants that are plankton. So instead, in the ocean, in the middle of the ocean, like we are here, I'm just going to turn so you can see that we're in the middle of nowhere here, and there's only water all around us. And the bottom of the ocean is very deep here. I don't know, probably several hundred meters at least. So when we're talking about the open ocean, like where we are today, the only plant that can live in here are tiny algae that are about, now we're talking about 50 micron in size maybe. And then, so they live in the surface of the ocean where there's sunlight that comes through. So those, instead of having trees in the middle of the ocean, we have these algae that are at the base of the food chain. So they're the food for all of the other organisms that live in the ocean, including the fish and the whales. So when we're talking, so we have the plants, so phytoplankton, which are the equivalent of the plants. And then we also have zooplankton. And zooplankton are a little bit bigger than the phytoplankton. And they're the direct food of many of the whales, the baleen whales that have kind of palms instead of teeth that can filter the plankton, and that's their main superfood source. So now we come to the third type of plankton that we have in the ocean, which is the bacteria, so the marine bacteria. So the marine bacteria are the recyclers. So if you think about garbage truck that's going around the town and then quietly collecting all of our debris and all of our food waste and so on, and the bacteria in the ocean, that's their role. And if there was no bacteria recycling the dead organic matter, there wouldn't be a continuity of the production in the ocean. I will summarize a little bit in French now. Dans les océans, le plancton, qui est l'équivalent des plantes sur les écosystèmes terrestres, dans des forêts, au lieu d'avoir des arbres, on a des toutes petites algues microscopiques qui sont à la base de tout l'écosystème, la chaîne alimentaire des océans. Donc, les plantes, des petites algues microscopiques, vivent dans la surface des océans, donc c'est très profond ici. Il y a seulement les algues peuvent vivre jusqu'à la profondeur qui est pénétrée par la lumière du soleil. Ensuite, on a le zooplancton, qui sont les tout petits animaux qui mangent le phytoplancton et puis qui aussi servent directement de nourriture pour toutes les baleines qui se nourrissent de plancton. Et puis aussi des poissons et d'autres organismes, d'autres animaux des océans. Ensuite, on a les bactéries. Et les bactéries jouent un rôle très important dans les océans parce que c'est eux qui permettent le recyclage de toute la matière organique qui a vécu et qui ensuite qui se décompose. Donc, c'est les bactéries dans les océans, c'est les décomposeurs qui font le recyclage des plantes qui meurent ou des poissons qui meurent. Donc, tout ça, le plancton est à la base de la vue qu'on retrouve dans les océans. Je pourrais aussi vous montrer comment on fait pour échantillonner le plancton. Alors, quand on est en pleine mer, comme ici, on a des instruments qu'on peut descendre dans la colonne d'eau. Et puis, c'est une façon spéciale pour collectionner ce plancton parce que c'est tout petit. Alors, on a besoin de... Je vais vous montrer. Je ne sais pas si vous pouvez voir ça, mais on a des filets à plancton. Alors, ça, c'est très grand. Vous voyez ici l'embouchure du filet, comme ça. Et puis, à l'embouchure, on a un petit... un instrument qui peut calculer combien d'eau passe à travers le filet. Alors ça, le filet, c'est comme un filet de pêche, sauf que la mèche est d'une taille très petite qui peut retenir toutes les planctons qui sont dans les océans. Alors vous voyez ici, comme c'est grand, c'est plus grand que moi en fait. Oups! Et voilà, et je vous montre comment... Donc, le filet, on le descend au fond de l'eau et puis on le retire rapidement. Et puis, tout le plancton se collectionne dans cette bouteille ici qui est au fond. Et puis, on peut dévisser et ensuite recueillir dans un flacon tout ce qu'on a ramassé avec cette bouteille ici au fond. Il y a comme des trous ici qui laissent passer ici. Donc, ça, c'est pour collectionner le zooplancton. Alors, je vais redire un peu en anglais pour résumer. So, the way we collect zooplankton is with plankton nets. And you can see earlier what I was doing. So this is a huge net that we lower at about 100 meters in the water column when the ship is stopped, and then we pull it. We pull it at a certain speed up and then to the deck, and then we collect all the plankton in this cup here at the bottom. It works. So it's hard. You can see here in this cup, so all the plankton is collected here. So this is similar to, whoops. So this is similar to a fish net, except that the mesh that we use is very small, and we can collect all the small organisms this way. Okay, I will stop here maybe and then take some questions.
Okay, Julie, just before we do that, I just have a couple little questions about the C3 for you. Are we still in leg 12, or has it flipped over to 13 yet?
We're still in leg 12.
Excellent. And how long have you been on the C3 for?
I've been on C3 for since September 10th.
Okay, excellent. And how much longer do you have to go? Are you on for the rest of the journey?
No, I'm off on September 30th.
Excellent. All right. Any highlights so far that stand out for you from the trip?
Yes, we had some great whale watching. One day we were out on the Zodiac on the hurricane, and then we just went out because we had to go sampling. It was late in the day, and then we came upon some humpback whales, and then so instead of sampling the plankton, we went and watched the whales. We did both in the end, but we watched the whales, and it was really nice. Some of them came about 10 feet away from the Zodiac.
All right, sounds amazing, and a very good reason to take a break.
Yeah. It's really amazing.
All right, excellent. Well, we have two museum groups joining us today. The first is from the Red Path Museum in Montreal, Quebec. Let me turn the microphone on. How's it going over there, guys?
It's great. We had a bilingual crowd and a lot of kids, and we really appreciated the explanations about zooplankton and the net that you showed us. Yep, it was a pleasure to see all of you, and I'm asking the kids here, do you have any questions on what you saw? Anyone? Do you have any questions about C3? What C3? Go ahead. Any questions? This is a ship that's going to the coast to coast, from Atlantic to the Arctic to the Pacific, and it has on board a group of scientists. And in front of you here is one of the scientists who explained a little bit about plankton. And there's also on board normal citizens like you and me. It's a great exhibition. They were here at the Red Path Museum a few months ago. We also had a live dive shown from Cambridge Bay. Do you have any questions at all? I can just comment. Yes, you. Pardon? Oui, la dernière fois, oui. Posez-moi une question en français, oui. L'air. Oh, est-ce que l'air c'est froid? Is the air cold?
L'air est maintenant on est en Alaska, on descend dans la côte de la Colombie-Britannique. Alors l'air s'est réchauffé depuis qu'on est parti de l'océan Arctique. Donc on est environ, je dirais à peu près 5 à 6 degrés dehors, centigrades. Et puis même l'eau devient de plus en plus chaude maintenant. Donc l'eau hier, quand on a échantillonné, c'était à 9 degrés centigrades. Donc c'est encore un peu froid pour se baigner, là, disons, aller sur la plage, tout ça, mais on a quand même réussi à faire du kayak hier en mettant des dry suits. Donc c'est une espèce d'habit qui nous protège contre l'eau, qui nous garde au sec quand on veut aller explorer en bateau ou soit en paddleboard.
Merci. Wait, do you have a question about C3? Go ahead. Anything. Yeah. Tu as une question? Vas-y. N'importe quoi.
Soyez pas gênés. Si vous avez des questions sur n'importe quoi, je suis prête à répondre.
Okay, well, we'll give them a minute to see if they have some more questions, but let's visit the Canadian Museum of Nature in Ottawa, Ontario, and let's see how they're doing.
Hi there, how are you doing? Merci beaucoup, Julie.
Bienvenue.
Bienvenue, excellent.
Plaisir.
Museum of Nature based in Ottawa. Yes. So we've had a few questions coming through, but specific to your field of work in plankton, we had a question here about storms, and it made me think that there must be a direct impact, is there not, on the populations of the plankton and the algae when there's a strong storm surge?
So when there's a strong storm, then there might be some water that's brought from the bottom, like deeper water that's brought to the surface, and that's important for bringing some nutrients to the surface. So yes, so that would be some influence of the storm. So after a storm, there might be more plankton that would grow if the nutrients are brought up to the surface.
Would there be more impact on zooplankton versus phytoplankton, and une différence entre les deux durant les orages?
So the question was what's the difference between the zooplankton and the phytoplankton?
In a storm scenario, where you were saying
storm scenario, yeah. Well, so the phytoplankton is usually the food for the zooplankton. So the two follow each other. So if you get a big bloom of phytoplankton, then usually you will get some of the animals that are going to come in. They're going to be there and grow and eat it up. So.
And has your research taken you up the food chain and to see the impact on the higher level animals that are feeding on the plankton and what could change in their numbers, the croissance de la population peut-être des poissons qui se nourrissent des zooplanctons après?
Okay, so one example of that was the other day when we were looking at the whales. When we were in a bay and there were these huge humpback whales, which is baleine à bosse en français. Alors, so we could see that they were chasing the plankton essentially. So what we did then is take an analysis of the plankton, the phytoplankton, so the little algae, and also of the zooplankton, and then we'll be able to assess what they were eating at that time that we saw them. So yes, it does affect. So the whales will follow the plankton, and then the birds follow the whales, and then so they know that's how they figure out how to find the food essentially.
Thank you very much. Let's see. We have audience questions at this moment. I'll pass it over to you.
All right, thank you. So let's just check one more time with Redpath in Montreal and see if they have a question for Julie.
Vous avez des questions pour Julie?
When you ever that
Pardon?
When you ever that
When? Oh, when? Oh, so the question, when will you arrive in the Arctic?
We already have been in the Arctic. So we were in the Arctic from September 10 to about September 15 or 16, I think, and then we were in Alaska. So the cruise, the leg that I'm on now, it started in Tuktoyaktuk, which is in the Northwest Territories. And then we went to the Yukon, we went to Herschel Island, And Herschel Island is an island where there is no, nobody lives on there now. It's basically a nature preserve, and it's an old whaling station. So there used to be whaling there, but now, as a result of climate change, there's a lot of erosion on the island. So all the people that were living there have been relocated in another area of the Northwest Territories. But it's a very interesting island to see all the leftovers from this era where there was a lot of whaling activities going on there. And after that, we went around to Alaska, so we were in the United States. And we stopped also in some northern communities there that were that also are making a subsistence living with some whale hunting. And one thing that was really interesting in those communities is you could really see the direct effect of the climate warming. And some of these indigenous nations, they put, after they hunt the whale, they put all of their catch, they put the whale meat and so on in the permafrost. So they have these sort of refrigerator or cooler or freezers, if you want, that are built in the permafrost. And these permafrost huts have started to erode and melt and then basically fall into the ocean. So we were able to see this firsthand, and that was interesting. And also, you know, it's going to have to lead to some adaptation for finding new places for these communities to store their frozen meat and so on.
All right, Julie. So being the weekend, we don't have any classrooms joining us today, but classrooms will watch the recorded version of this during the following week. So I'm going to ask you a few questions, just a little bit about the science that classrooms might be interested in. So building off what you were just talking about with the global warming, have you seen an impact on the smaller community of phytoplankton and zooplankton due to global warming?
Well, part of the work that we're doing now on this cruise is to get a survey of what's living in the water right now. So especially the bacteria and the plankton and so on. So we're collecting water samples all along the cruise, almost every day in all the locations that we come to. And so afterwards, after the cruise, we will analyze these samples. And what we're going to do is extract DNA. So you know that DNA is is the code for life. So basically, all of our proteins and enzymes and everything that makes our cells work is encoded in this DNA. And so it's possible to get a barcode for different organisms, and this is what we're going to do after the cruise. We're going to basically barcode everything that we saw on this trip, and this will serve then as a baseline for what's the state of affairs in the ocean right now. And so, because it's never been done before along this, it's such a wide scope along the Arctic, the Canadian Arctic. So this is going to be our baseline, and then realizing that this baseline is already changed, right? Because the climate change is going to be going on for a little while, but at least we'll have the baseline for 2017, and then in 2 years or 10 years from now, then we'll be able to better assess what's changing.
Yes, we often talk about this. The Canada C3 is going to leave behind quite an amazing legacy of science, going coast to coast to coast like this. And for years to come, I'm sure there'll be a lot of unpacking of the data that does come from this expedition.
That's right. And also, so the DNA, the samples that we're collecting and we'll be extracting, we'll be archiving this DNA. And DNA has a property that it can last a really long time. So it's not, it's if you put it in the freezer, it will last for years. So in years from now, if we want to go back to these samples, we will be able to do that. So it's like it's kind of a little bit like a museum collection. So it will be the museum collection for the microbes and the DNA samples. And there's also some people who are working on environmental DNA. So they, it turns out that when you have a fish or a whale that swims in the ocean, they probably shed a few cells. as they swim along. And our sampling methods can collect these cells. And so there's a possibility that we'll be able also to detect them in our DNA samples.
All right, excellent. So the museum, Redpath Museum, just sent me a couple questions via text, but let me see if they want to ask them live. So let me turn the microphone on. You guys had a great question about the permafrost. Do you guys want to ask that question?
Well, the person who asked the question is not here, but I can read it to you. Do the permafrost have greenhouse gases trapped in them? That's the first question, like the permafrost that you talked about. They've heard in Siberia they have methane trapped in the permafrost, but what about the permafrost that you observed? Was there any greenhouse gases trapped underneath?
Yes, so there will be some greenhouse gases trapped in the permafrost, and then it's the same as in Siberia. So if it melts, there will be some methane that's going to be released. Now, it's interesting that you're asking about the methane because the methane is produced by microbes or by bacteria, and so there's usually two types of bacteria that work together. There's a bacteria that produces the methane, and there's a bacteria that breaks it down. So when things are not out of control, then these two bacteria work together and they produce and consume the methane. But if all of a sudden the temperature warms up and then everything melts, then the bacteria that degrade the methane might not be able to keep up with the methane production. So yes, so that's a concern. So there's a boy the boy there had a question. Oh, at the Redpath Museum.
Oh, let me turn his mic on. There we go.
Oh, qu'est-ce que vous mangez? Like what?
Qu'est-ce qu'on mange sur le bateau?
Yeah, oui, c'est ça. What do you eat?
Ok. So, qu'est-ce qu'on mange sur le bateau? Oh, on a une fantastique cuisinière qui s'appelle Gail. Et puis, on mange toutes sortes de choses. On mange, il y a de la nourriture pour les personnes qui veulent manger des mets végétariens. Et il y en a pour ceux qui veulent manger de la viande aussi. Alors, on a mangé du saumon. Ok, je vais te dire ça. Donc, le matin, on a des œufs. Et puis, on a des omelettes. Et on a du pain doré, des gaufres. Et puis, bon, du jus d'orange, toutes sortes de choses, comme tu mangerais chez toi à la maison. Ensuite, le midi, ça dépend si on est sur le bateau. Là, on mange des mets chauds. Donc, de la viande, des pommes de terre, des légumes. Et puis, si on est en excursion sur le terrain, donc si on va sur la terre, sur le bateau, là, on apporte un dîner qui est déjà préparé, comme des sandwichs ou des barres de fruits. Et puis, le soir, ben, c'est un peu la même chose. On mange des pâtes, du spaghetti, des choses comme ça. Puis, de temps à autre, on a un gros buffet. Donc, on s'assoit dans une grande salle avec des grandes tables. Puis là, on a un repas spécial qui a été préparé par soit des personnes qui sont participants sur le bateau ou avec l'aide de la cuisinière. Donc, on mange très, très bien sur le bateau. Mais il ne faut pas avoir le mal de mer parce que quand on a le mal de mer, on n'a pas très faim.
All right. Well, question about your sampling methods, Julie. You talked about dropping the net down to about 100 meters and then slowly bringing it up. Why that method as opposed to, say, dragging it behind a smaller boat like the Zodiac?
Parce que, disons que quand on le met jusqu'à 100 mètres et puis on le retire vers le haut, à ce moment-là, on a une intégration de la colonne d'eau. Should I speak in English now? So we have an integration of the water column all along. So we want 100 meters. It's on the shelf. It's usually about 100 to 200 meters, and then so we can measure all. We integrate what's in the water column that way, as opposed to towing. Towing, you would just get the surface. So by going from deep to surface, then we get an integration of what's in the water column. Because at different levels in the ocean, there will be different types of plankton. So we want to collect it out. And I have another thing that I can show you as well for the water samples, because the net that I showed you, it's very small mesh, but it still would let some of the algae through, and the bacteria certainly don't get collected by these nets. So I'll show you one more thing that I have. And this is called a Niskin bottle. Can you see this? It's a Niskin bottle. And then, so the way this bottle works is we send it down like this, but this caps at both ends. So you can see here I have one cap, so I can open this, and then I open the bottom like this. And when I send the bottle down, it's open, so the water is flowing through as I take it down. And we have a little weight that we send on it. So this bottle is hooked up to a wire, right? Because you can't just put it in the water like this. So it's hooked up to a winch and to a wire. And then when it's open like this, we can send it down. If it was capped, then it would be floating. So that wouldn't work. So and then the water flow, and so we send this bottle to the depth that we want to collect the water at, and then we send a messenger, something called a messenger, and oops, something like that. And it clicks on a button that's here, and then both the top and the bottom are closed like this. And I have the bottle upside down, so it would be like this. And then we bring it up, and then we have a water sample that would be collected, let's say at 100 meters instead of like the surface. So this is the kind of instrument that we use to collect the bacteria and the phytoplankton as well.
Well, thank you for sharing with us. I'm going to check in at the Canadian Museum of Nature because I thought I saw someone near the camera. Let's see if they have another question for us in Ottawa.
Well, we've been talking to a lovely young boy, and he's gone here, and we have lots of questions about how that net worked. I was wondering if Dr. Judy could bring back the net that had the cup at the end, because he didn't get a chance to see that net.
There we go. So here's the net. So okay, here's the mouth of the net. So this is hooked up to a big hook, and this is hooked up to a wire that we can lower down in the ocean. So it just goes down like this, right? And you want to see the cod end, so it's very long, so I can't really show all of this in the screen that I have here. So this is where the plankton is collected at the end. So,
great.
Do you have a question?
Kyle's over here. He was very happy to see the net.
Okay, and then, oh yeah, so.
Yes, go ahead.
So at the end of the net here, at the end of the net, there's like a long rope, and then we attach a weight here because, you know, in the water, it needs to be heavy to sink. So we attach a big lead weight at the end here, and this goes down first, and then after that there's the cod end that goes down, and then there's the rest of the net like this. And then so when we pull it up, then it's all in one big long sweeping net here, and then this mouth here collects all the plankton and concentrates it as the net is pulled up, and the water flows through here through the mesh where the plankton is collected.
Thank you so much. I have one more question. How many days at sea have you been, or will you be, without seeing land?
Oh yes, so for this leg we had a lot of days at sea because we're going around Alaska, and then there's not a lot of communities that we can stop at, and then so we're going mainly through the Canadian communities as well. And so going through the Bering Strait, we went through this chain of volcanic islands called the LaRoche Islands, and that was really great, a great place to go through. But you know, like nevertheless, there's not a lot of coastline, so we have to go in the middle of the ocean here. And the same thing, leaving. So yesterday we were in Kodiak, which is in Alaska. It's also volcanic islands there. And then, so now we have to go to the coast of British Columbia, and we have to go in the ocean. So we had, I would say, about 6 or 7 days at sea so far. Yeah, and sometimes it was a little bit rough, so it's hard to, you know, keep your balance. So like last night, the ship was quite rolly because depending on where you want to go in the ocean, you have to direct your boat in the ship in that direction. And, you know, sometimes the waves are coming sideways. So the ship was very rolly. And this morning when I woke up, I had my book and my radio here, and my hairbrush were in my garbage bin. So it flew off from my little table. So, but that's okay. There was nothing dirty in my garbage bin. So.
I hope it wasn't the big buffet dinner last night as well.
I'm sorry? I didn't hear that.
J'espère que vous n'avez pas mangé un gros buffet le soir et avant.
All right. Well, Julie, I have another question for you about the phytoplankton. So we're talking today about how small they are and how they have a big impact. And you talked about how they are the basis for the food chain. But I want to ask you about something else they do. So we know trees get a lot of credit for oxygen production. Can you tell us a little bit about the little organisms in the ocean and how they can help with that too?
Okay, so that's a very good question. So the phytoplankton in the ocean, so there's algae like diatoms. You know, diatoms they make little silica shells, and then they're enclosed in like a little pill box, or like two parts that come together like this. And so when these algae bloom, and in general, the phytoplankton, they make 40% of the oxygen that we breathe. So they're very important in the balance, keeping the balance of the oxygen in the atmosphere. So does that answer the question?
No, excellent. Yeah, no, it's. I think it's amazing, these little creatures that we rarely think about or see in our oceans, but you know, without them forming the basis of the food chain and trapping carbon and producing large amounts of oxygen, they actually are powerhouses, these little microorganisms, when they're all pooled together doing their thing.
That's right. So so seasonally you get blooms in various areas of the world. So in the Arctic we get the bloom, and so March, April. No, I mean in the northern hemisphere we get the bloom in March, April, May. And then in the southern hemisphere, it's a different season. But yeah, so there's like, especially in the coastal areas, you see seasonal plankton blooms or phytoplankton blooms that are then the beginning of the food chain for that year. So, and then they produce oxygen.
All right, excellent. Well, let's visit Redpath Museum one more time.
Hi.
Hi.
So this is a great event, a lot of questions. We also want to show you like an activity kids were busy with. It's called an educational game developed by the Faculty of Education called Tree of Life. It's by playing creatively and imaginatively. Participants, who can be kids, it can be family members, even grandparents, all included around the table. And they get to discover different aspects of you know, biodiversity. So just wanted to share that. And there are some new members in the audience. They are discovering what C3 is, and perhaps they have a question or two on research or anything simple about what's going on in C3. Do you have a question? En français, tu as une question? Oui. Well, I do have a question. Like, how many people are traveling on board at one time in the ship?
Est-ce que c'est pour répondre en français ou en anglais?
Combien de personnes voyagent dans votre bateau à n'importe quel moment, par exemple?
Je pense que présentement, on est 28 participants. Ça dépend, il y a environ 50 personnes, je pense, sur le bateau, incluant les membres de l'équipage. Donc, on a un capitaine Stéphane Guy, qui est très cool. Et puis, bon, les membres de l'équipage. Et ensuite, il y a toutes sortes de personnes ici. Il y a 2 ambassadeurs de jeunesse. Donc, une jeune fille qui vient de Tokiaktuk, qui est de nation indigène. Ensuite, on a des gens de l'île du Prince-Édouard, 2 personnes de l'île du Prince-Édouard. Moi, je viens de la Nouvelle-Écosse, donc Halifax. Et puis, donc, il y en a plusieurs qui viennent de l'Alberta. Donc, à bord aussi, on a 4 scientifiques. Donc, il y a moi qui travaille sur le plancton. Il y a une personne qui travaille sur les ours bruns et puis les ours polaires. Ensuite, il y a une personne qui travaille sur les plastiques dans les océans, les microplastiques. Et puis, il y a une personne qui travaille au département de pêche et océan à Victoria et qui collectionne, qui nous aide à collectionner tous les échantillons qu'on doit ramasser. Aussi, sur le bateau, quand on va sur la Terre, on ramasse des échantillons pour les plantes et puis on fait des observations sur les mammifères et tout ça. Donc, il n'y a pas seulement l'échantillonnage des eaux, il y a l'échantillonnage aussi pour faire des collections de plantes et puis d'autres, de lichens. Il y a d'autres projets scientifiques aussi.
D'accord, merci. C'est une très bonne réponse. Et il y a d'autres questions? Oui, il y a le site web de voir tous les détails. Comme elle dit, elle a une équipage de scientifiques et aussi des citoyens normaux pour commémorer la 150e anniversaire du Canada. Ils traversent de la côte atlantique à Arctique. Maintenant, ils sont en dans l'ouest. Oui.
Il y a toutes sortes, il y a aussi des écrivains, il y a des personnes, il y a 4 personnes de Première Nation, il y a des journalistes, des musiciens, on a 2 musiciens à bord qui nous donnent des concerts, dépendant de nos activités le soir. Et puis, on a des avocats, des personnes qui travaillent qui font du travail social chez eux, pas sur le bateau, mais à la maison. Donc, c'est toute une participation, c'est très diverse, mais les personnes qui sont sur le bateau ont été choisies parmi beaucoup qui ont fait de demandes pour embarquer sur le bateau. Spécifiquement, les personnes qui sont pour les ambassadeurs de jeunesse ont été choisies parmi 3000 participants. Donc, ils sont vraiment spéciaux.
Avez-vous remarqué des espèces que vous n'avez pas vues ou c'est tout était comme si déjà familier quand vous avez fait des plongeons ou des choses, quand vous avez fait vos samplings?
OK. Donc, jusqu'à maintenant, on n'a pas regardé ce qu'il y a dans les échantillons du plancton parce qu'on a besoin de microscopes et on a besoin de faire des extractions de l'ADN. Et puis, à partir de l'ADN, on va pouvoir faire regarder quelles espèces on a dans le plancton. Donc, le truc avec les bactéries et puis les phytoplanctons, c'est qu'on n'a pas encore découvert toutes les espèces qui habitent les océans. Donc, c'est très, très, très divers. Donc, tu peux penser donc, la différence entre un zèbre ou un poisson et un humain est beaucoup plus petite que toutes les différences qu'on voit entre certaines espèces de bactéries. Donc, au point de vue, disons que métabolique, disons que les différents types de bactéries, ce qui leur sert de nourriture à eux, sont les nutriments qui leur servent. Donc, c'est très, très divers en fait. Donc, s'il y a un produit chimique sur, disons, qui est dans une molécule chimique qui est dans les océans, c'est sûr qu'il y a une bactérie qui va être capable de l'utiliser. Donc, on n'a pas encore, une complète vue de toute la diversité qu'il y a dans les océans sur le côté des bactéries. Non, maintenant, sur le côté des mammifères marins, tout ça, là, on n'a rien découvert jusqu'à maintenant. Et puis, sur le côté de l'inventaire des plantes qu'on est en train de faire, là, à chaque fois qu'on va sur le terrain, sur la terre ferme, là, le but de cet exercice, c'est un peu de voir, est-ce qu'il y a des espèces qui migrent vers le nord, donc des espèces qu'on voit dans le sud du pays qui commencent à migrer vers les endroits plus nordiques. Ça serait comme une étendue de leur diversité, de leur distribution, là, vers le nord à mesure que le climat se réchauffe. Donc, non, jusqu'à maintenant, on n'a pas découvert de nouvelles espèces de plantes et puis d'animaux, disons de mammifères, de poissons, mais c'est aussi pour regarder comment les animaux sont dispersés dans les océans.
Oh, merci beaucoup pour votre réponse. Mais quand l'équipage de C3 était au musée de Redpath, quelqu'un a parlé par exemple, le réchauffement climatique qui a changé les territoires des animaux. Et comme par exemple l'ours polaire, maintenant on peut rencontrer le grizzly. Et ils ont maintenant des espèces hybrides comme le pisli. On a vu une photo à l'exhibition à Biosphère. Et je me demande, est-ce que pendant votre trajet, est-ce que vous avez eu l'occasion de voir quelque chose du bateau? C'est peut-être trop loin, vous avez quelque chose, mais vous êtes au courant des changements de territoire des animaux? D'accord.
Oui, c'est ça, donc un peu, mais jusqu'à maintenant, on n'a pas découvert de nouvelles espèces, sauf comme vous dites, il y a les hybrides des ours polaires avec les grizzlies. Ça, il y en a quelques sur le bateau qui travaillent là-dessus. Mais non, jusqu'à maintenant, on n'a rien découvert dans ce genre de choses. Mais ça, l'exemple avec l'ours polaire, c'est un très bon exemple qui est visible. Ça, on peut voir les traits de l'ours polaire et du grizzly qui sont intégrés dans la nouvelle, dans l'hybride. Mais en fait, ça peut aussi se passer pour d'autres, peut-être des espèces de plantes ou d'autres types d'animaux. Mais jusqu'à maintenant, j'ai pas entendu parler que ça a été découvert. Et puis, on a cherché des ours quand on était à Kodiak, parce que Kodiak, il y a le fameux Kodiak Bear, qui est l'ours brun, qui est aussi un grizzly. Mais comme on n'en a pas vu, on n'a pas eu la chance d'en voir.
All right. Thank you very much, Julie. We will expand on that a little bit more on the 26th. We'll be connecting for our next Canada C3 Hangout with Mark Edwards, and he'll be talking a little bit about bears in the Arctic. So we're looking forward to that on the 26th at 1 p.m. Eastern. So please join us for that one. And let's check in just quickly one last time with the Museum of Nature, and then we'll sign off for today.
Okay. Thanks, Joe. Thanks, Julie. I think all of our questions here we've had answered, and we've had many passing visitors at the museum who have enjoyed listening to Julie. So thank you so much, and we'll be very excited as well to tune in on the 26th to learn more about the bears research. Thank you so much, C3. Safe voyage. Bon voyage.
All right.
Okay,
thank you very much. On the 28th as well of September, we'll have another hangout with some of the youth ambassadors from the C3, and that's at 1 o'clock Eastern as well. Julie, thank you so much for hanging out with us today and sharing your experiences from the C3 so far, as well as some of the work you're doing with some overlooked organisms in our ocean.
Thank you. You're welcome.
All right, Redpath Museum, thank you so much for hanging out with us today. Canadian Museum of Nature, thank you so much. And we're going to sign off for today.